Okay, this video is the second video in the photosynthesis series of videos. This is actually a very primitive diagram of a Bohr model of an atom, and if you've had chemistry, you know that this isn't an exactly accurate model of the atom, but it's a pretty decent model for us to use to kind of discuss the basics of what are going on. So, atoms are capable of absorbing and emitting certain frequencies of light energy. So in this case, you're seeing electrons jumping from lower states of energy to higher energy levels and giving off energy and falling back down to the ground state. And they emit the exact same amount of light energy that they have absorbed. So chlorophyll molecules can capture light energy and pass it along to each other in a really similar manner to this. Accessory pigments can capture additional wavelengths of light that chlorophyll can't, but they're also used to protect chlorophyll from damage by capturing an excess high energy light and safely dispersing it in the form of heat. So sometimes a photon of light can impart enough energy for an electron to leave an atom entirely. And this is what we're going to be discussing today when we talk about the light reactions. And in a previous video, we talked about there being a slight difference between chlorophyll A and chlorophyll B. And in this video, we're going to talk exactly about the difference between chlorophyll A molecules in terms of what their job is in the process of photosynthesis. So before we get into the details of the light reaction, we have to figure out where we are in a plant and in an organelle called the chloroplast. So first of all, if we're in a leaf, we're most likely in the palisade layer. It is not the only layer of the leaf where photosynthesis takes place, but it is the layer that has the most chloroplasts in the cell, and it's towards the top of the leaf, which gives it really good access to light. So we're probably in one of these palisade cells. We could be in an algae cell, of course, and we could also be in many other types of, we could be in the spongy mesophyll, but statistically speaking, we're probably in a palisade cell. And now we're going to go into a cell and into a chloroplast. And now we're going to look at a cross-section of chloroplast, and we're going to be going into a granum, which is a stack of thylakoids, and we're going to look at 1/4 of a thylakoid. So this is actually a cross-section where we've sliced a thylakoid open, and we've zoomed in on half of it so that it's large enough for you to see what's going on. You probably recognize what these are. These are phospholipids, and it's a phospholipid bilayer. We learned about them in the previous 2 units. The heads are hydrophilic and attracted to water, and the tails are hydrophobic and repelled from water. And so if you've got a watery environment like a cell, and you have enough phospholipids, they will flip away from each other so that the heads are on the outer surface and the tails are away from either surface where there's a lot of water. So if we look at this diagram, we have actually one copy of each part of this system that I'm going to talk about. But in reality, there will be multiple copies of everything I'm about to show you. But just to kind of orient you to where we are, this is the thylakoid membrane. The inside of the thylakoid membrane is referred to as the lumen, and the outside is the stroma. And the stroma is where the second phase of photosynthesis takes place, and that part is called the Calvin cycle. So if you look at these little collections of green circles, those are representing the pigment molecules. They are embedded in the phospholipid bilayer. Some of them are chlorophyll A, some of them are chlorophyll B, and some of them are accessory pigments. They're like a team that works together. So PS2 stands for photosystem 2. And we're going to discuss photosystem 2 before photosystem 1. This always bothered me as a student. But actually, photosystem 1 is referred to as photosystem 1 because it evolved earlier in evolutionary history, not because any of what goes on happens there first. So just keep in mind that even though we're starting with photosystem 2, and there is a logical progression, but that what happens at photosystem 1 and photosystem 2 actually happens simultaneously. I just can't talk about 2 things simultaneously. So a photosystem is a group of pigments. Now, we learned in the previous video that red and blue wavelengths of light are the ones that are most absorbed, which is why we show you sunlight as a rainbow, but the blue and the red are the ones that are being absorbed. Now, each pigment molecule absorbs a little bit of energy, but they donate all of that energy to a special chlorophyll A molecule called the reaction center. And you can see it blinking right here, yellow. Students always want to know what's so special about this chlorophyll A molecule. Nothing structurally, but what's important about it is its location being right next to this enzyme right here, this blue enzyme. If you take advanced placement or AP biology, they'll tell you the names of more of these molecules, but I'm going to keep it pretty basic here. So this chlorophyll A molecule, and yes, the reaction center is always a chlorophyll A molecule, and never a chlorophyll B molecule, is going to lose electrons. But first, students always want to know, well, what about chlorophyll B and chlorophyll A? Well, the rest of these pigments in the photosystem, some of them are chlorophyll A and some of them are chlorophyll B. So while chlorophyll A seems like it's the most important, you need a lot of chlorophyll A and chlorophyll B to make this work, to collect enough energy for the next thing that's going to happen. So the next thing that's going to happen, once they've collected all of that energy from the sunlight, is electrons are going to be kicked out of that chlorophyll A molecule that we refer to as the reaction center. But those electrons have to be replaced because a chlorophyll molecule can't keep losing electrons over and over again. It's harder to remove more and more electrons, so they have to be replaced. So what replaces them is this enzyme that actually pulls electrons from the hydrogen atoms in a water molecule. So if you look here, it says H2O, electron donor. And the reason that you get hydrogen ions is because you're pulling 2 electrons, one from each hydrogen atom, which leaves them as ions, H+ ions. And what you have left over is 1/2 of an oxygen molecule. Students always get very confused. Well, why is it 1/2 of an O2? Well, if you look at an H2O, there's only 1 oxygen atom. So in order to get O2, you would actually have to do this twice and rip apart 2 water molecules. But fortunately, that happens really quickly. So you can do that. So 2 water molecules are actually pulled apart, and you really get 4 hydrogen ions and an O2. Okay? Now, once you have these electrons, they have extra energy. And if you ever look at a biology diagram and there's like a zigzag line around something that kind of looks like lightning, that tells you there's some extra energy there. And the energy from those electrons is going to be used to do something really important. It's going to be used to pump hydrogen ions into the lumen against the concentration gradient. You should know by now that if you're moving ions from low to high concentration, that's going to require energy. And in the past, you've been told that active transport always uses ATP. And in this case, the energy didn't come from ATP, it came from sunlight. But if you look here, we have more hydrogen on the inside of the lumen and hydrogen ions being pumped inside. This purple molecule is referred to as a proton pump. And what you've seen is active transport because we've gone from low to high concentration. And remember, you also learned in the previous unit that ions can't go right through the phospholipids. So they can't diffuse right back out. That's going to be important in a minute or two. Now, this purple molecule that pumped hydrogen into the lumen is referred to as a proton pump. Sometimes students wonder why. And they're always asking, well, why isn't it called the hydrogen ion pump? Well, that's really the same thing because a typical hydrogen atom has 1 proton and 1 electron. If you have pulled the electrons off of it, like right here, what's left is a proton. Of course, there might be a neutron there, but people don't really talk about neutrons unless you're talking about nuclear chemistry. So for now, we don't care about the neutrons. Now we care about the fact that we have hydrogen ions. So now we have a high hydrogen ion concentration inside the lumen. The next thing that's going to happen is facilitated diffusion, which is a type of passive transport. And those hydrogen ions are going to flow through a molecule called ATP synthase. Sometimes books will call it ATP synthetase. If you come across that, that's just an older version of the name for this enzyme. You should be able to tell that it's an enzyme because it ends in the ACE ending, and you should be able to tell what it does because its ATP synthase, that tells you it's going to synthesize ATP. So the hydrogen ions are going to flow through that protein channel. That is a protein channel and is also an enzyme. Now, oftentimes students get confused. Well, how can it be a protein channel and an enzyme? Well, it is. The flow of hydrogen ions through this kind of act like a water wheel, taking the energy, the kinetic energy of the flow of hydrogen through that. And there's a mechanism in this molecule that actually spins and pushes together an ADP molecule with a third phosphate to make an ATP molecule. Students often think that the hydrogen ion ends up in the ATP molecule. It doesn't. The only purpose is for the hydrogen ions to flow through that red ATP synthase in order to create the kinetic energy to convert into chemical energy. So if we look at all of the energy conversions, it's really interesting here. First of all, we had solar energy that created electrical energy, and it created an electrical potential energy because you had a whole bunch of hydrogen ions on the inside of the lumen, which makes the inside of the lumen relatively positively charged compared to the outside. Because these have the same charges, they're repelled from each other and are going to tend to want to diffuse out of the lumen. They can't go right through phospholipids. We learned about that in the previous unit. And because they can't, they have to go through facilitated diffusion. So they go right through this ATP synthase, and we have ATP. So now we have our first product of the light reactions that are going to be needed for phase 2, which is the Calvin cycle. Now, students often want to know, well, where does the ADP come from to be combined with the inorganic phosphate? Every time the cell uses ATP for anything, the third phosphate comes off and you get ADP back and inorganic phosphate back. So there is a recycling going on. Once it's used for the Calvin cycle, you get ADP back. Now, at the same time that the photosystem 2 was collecting energy, photosystem 1 is also collecting energy from the red and blue wavelengths of sun, and they are pooling it together, donating it to that chlorophyll A molecule in the reaction center of photosystem 1. But this time the energy is being used for something else. The energy is being used to combine an NADP plus hydrogen to make NADPH, which is going to be used as an energy carrier to give energy to make sugar in the Calvin cycle, but also to donate hydrogen. Remember that carbon dioxide has carbon and oxygen. Carbohydrates have carbon, oxygen, and hydrogen. So this molecule is going to provide the hydrogen. Where did it get that hydrogen? If you go back far enough, from water. So remember when hydrogen was pulled off of water? Eventually in the Calvin cycle, it's going to go back together to form some water, but it ends up in the carbohydrates. So once NADPH is used in the Calvin cycle, we get NADP plus back to come in and get more hydrogen and make more NADPH. So this is recycled after use. It's important to remember that while all this is going on with the light reactions, the Calvin cycle is actually happening out in the stroma. So this NADPH and ATP will be used producing NADP plus and ADP that can come back and pick up a third phosphate and more energy, and hydrogen and more energy. So again, the energy from these electrons is used to make NADPH, and that was collected by photosystem 1. Now, remember how we said that the electrons in this reaction center had to be replaced by the electrons taken from water? Well, these electrons are passed along, and notice here, after the hydrogen ions have been pumped into the lumen, you still have electrons, but they don't have the extra energy anymore. Once the electrons are lost from this chlorophyll A molecule in the reaction center in photosystem 1, these electrons replace the electrons lost from the chlorophyll A molecule in the reaction center of photosystem 1. So if you follow electrons through their whole history, they originally come from water, replace the electrons lost from the chlorophyll A molecule in the reaction center in photosystem 2, have extra energy, which is used to pump hydrogen ions against the concentration gradient into the lumen. Then those electrons replace the electrons lost from the chlorophyll A molecule in the reaction center in photosystem 1, And then they get energized again. The energy that's picked up from photosystem 1 is used to produce NADPH. Now, all along, this is creating this high concentration of hydrogen ions on the inside. Doesn't reach equilibrium during the day as long as there's sunlight. And because you have more hydrogen ions on the inside than the outside, it's going to constantly be diffusing through ATP synthase, constantly producing ATP all day long. So, the primary pigment used by plants to capture light energy is chlorophyll A. These reactions occur within the thylakoid membranes, and photosystem 2 starts off the process of photosynthesis. The movement of electrons from photosystem 2 to photosystem 1 drives a proton pump moving hydrogen ions from low to high concentration. This is an example of active transport. This gradient of hydrogen ions is then allowed to flow from high to low concentration, also called passive transport, and more specifically, it's called facilitated diffusion. This results in the production of ATP. Photosystem 1 collects more light energy and drives out an electron, which is used to synthesize NADPH. The electrons are continually being replaced by the electron donor, water. So this is why we need to water our plants.